So we're going to have our panel next, and James Lendemer is going to be back with us, and as is Sharifa Crandall and Carolee Bull, who is standing in for John Pecchia. So thank you, Carolee, for doing that on short notice. The first thing I'm going to do is let the panelists do an introduction of themselves and a little bit about their research. So Sharifa, I'd like to start with you.
So hello, everybody. Oh, I also played trumpet once upon a time, so I can project, but I will use this. So hello, everybody. My name is Sharifa Crandall, and I'm an assistant professor in the plant pathology and environmental microbiology department. And I have a background in fungal ecology and plant pathology. And so currently my lab is about soil-borne pathogens. And I also work with those elusive endophytes. Hopefully we have a chance to talk a little bit more about them today. And I think that's good.
So my name is Carolee Bull, and I serve as the head of the Department of Plant Pathology and Environmental Microbiology that you've all heard so much about today. And I work closely with John Pecchia, who is the director of the mushroom research facility on campus. It's a unique facility. There are only 2 publicly funded facilities like this in the world, and this is really an amazing facility. The kind of work that has come out of there has helped to make, in fact, the Pennsylvania mushroom industry leaders in the country as well as in the world in many ways. They're primarily working with agaricus production, but John is very interested in exotic mushrooms as well. And yeah, I think that's maybe what I'll say about that. The organization is funded by both endowments from the mushroom industry, but also I think since 2014, as a group, we've had multi-million-dollar grants continuously. And so we just wrote another one last weekend. And so let's hope that one gets funded. My particular role in working with the mushroom industry or with mushrooms is I'm a phytobacteriologist. I work on diseases caused by bacteria on mushrooms or plants. In particular, I do a little bit of bacterial taxonomy, and what I say is translational taxonomy, understanding what the organisms are, and then using that information to strategize management strategies based on their ecology and other factors.
All right, thank you, Sharifa and Carolee. James, we've already met you, but would you like to say a word or two about your path, maybe?
Sure. So I'm James Lendemer. I am an associate curator here at the New York Botanical Garden and assistant professor at the City University of New York. And so my research mostly focuses on sort of biodiversity and ecology in all its forms of lichens. And really, I've spent my career trying to understand what they are, why they are there, and why they are and are not where they are. And really in my work and my students' work and that of my collaborators has really, runs the gamut from everything from population genetics and genomics to descriptive taxonomy and ecology, macroecology, all sorts of things. I'm a person with the lichen expertise and sort of I work with other people who know things about other things to make the world a better place.
Great. James, I'm just going to do a little PR thing for you right here, right now. Do you want to say about among your many works, you wrote a book called We did. You're right. I was going to have him sign it, but he's gone. Oh, right. Oh, good. Yes. Excellent. So James, I've brought a copy of your book and it's in the back of the room. And I was going to ask you to sign it, but then I'll have to maybe. Excellent. Thank you so much. Yay. Okay. Great. So I am a microbiologist in the geoscience department. And so that's one of my hats, and my other hat is as the Ecology Institute director. So I'm bringing both of my hats today. in addition to a third hat, which is that I direct the Astrobiology Center on campus. And so I am, in many ways, I think of myself as a planetary ecologist, hopefully not the dying type, if you've been watching Dune lately, in which a planetary ecologist famously dies. Anyway, I feel perfectly safe in my role as planetary ecologist. So what we want to do here in this panel is continue to be very freewheeling. And so I'm asking the audience to participate by asking questions. I do have a bunch of questions here, but I'd much rather have the questions come from you all on the Zoom and in the room. And so I think one of the goals that I have for this panel is to try to assess the promise and also the challenges associated with using fungi in association or combination with other organisms. And so we're really focusing on these interactions that could remove frictions or add frictions. And we're starting from this place of reductionist science where we like to have things in isolation, including microbial cultures. And but we now have these new tools for interrogating how biological systems work without isolating the components first. And so we really want to talk about progress and the promise and the challenges of doing that.
Okay, so I actually had a few words, and so I know it's been a long day, and so I'll keep this very concise. Sometimes I think it's nice to maybe have like 3 take-home messages, maybe, at least from me. I don't know. I'm sure there are a lot that you can take home today. And so this has to do with when you think about fungi and their ecology. Okay, and when you're thinking about making those links between fungi and biomaterials. And so sort of these 3 things are sort of a framework, or at least my framework of how I think about it. An example, and then making a link or multiple links. Okay, so the first take home, the framework. So a lot of my work with fungal ecology is trying to understand fungi across time and space, and in particular, their biology and their traits. And I'm sure you've all studied, traits in general about maybe humans before or other organisms, but fungal traits in particular, I think are important within as a framework. When you're trying to think about biomaterials downstream. So what are fungal traits? Does anybody know or have any ideas from the audience, maybe? What's a fungal trait? Yeah. That it procreates and spread. Absolutely. Dispersal, reproduction. Absolutely. Traits is a broad term, right? Yeah. O's answer on Zoom. What is it? Absolutely. Sometimes it's nice to think about these traits as phenotypic, or, maybe the color of spores that allow them to deal with, lots of UV light, right? They actually have melanin, believe it or not, just like us, okay, in our skin. So do fungi. And so it's very interesting to think about traits, because when you have those genetic traits, you can also study those, and you can study maybe their function as well, right? So again, just to think for yourselves, if you're interested in biomaterials, sort of what is it that you're interested in downstream, and what are those functions, right? That, James was alluding to earlier, that are sort of ubiquitous among certain types of fungi, perhaps closely related fungi. Some fungi are very good, some, not most, but some are very good at degrading lignin, for example, right? Breaking down wood. If it wasn't for that trait, we'd be knee deep, okay, in leaf litter and logs, but the fungi help us with that. Okay, so that's that framework. So just think about those traits, some of which are functional traits, genotypic, phenotypic. Okay, so what could be an example? Because remember, this conference is also about those interactions between organisms. It's not just about fungi sort of in a vacuum. Okay, so here comes at least a great example in my mind, because it's something I'm working on with a bunch of colleagues here at Penn State. And it's this idea of endophytes. Okay, so what is an endophyte? Does anybody have an idea? No? Okay, that's okay. It's okay if you don't know what we've probably heard this word kicked around a little, at least today, right? Yes, and people on Zoom are probably like, yep. Okay, so in this room, we actually have a real expert on endophytes, you know, Dr. Maria Del Mar Jimenez Gasco. She's in the back. So if you have any questions, she's been thinking about endophytes for years, and her research is really on that. And I'm just beginning to do this in my career. But endophytes are fungal endophytes are fungi. Endo means inside. Phyte typically means plant, but we'll also say they can also live inside of, they're like endosymbionts, can also live inside a lichen too. Fungal endophytes, okay, are so interesting because they have so many functions that, oh, and by the way, they typically live inside a plant, but they typically do not cause harm. Okay, typically do not. An endophyte can transform into a pathogen. And that's interesting too, right? So to wrap this up, because I don't want to take too much time, I think when you think about an example of like maybe where we can think about functions, these fungal endophytes, some people have shown, you know, and there's actual research here in the plant literature, for example, that fungal endophytes help deter herbivory. That's a big deal if you're thinking about crops, right? And maybe you could select for endophytes that live inside a corn leaf or a strawberry, maybe, I don't know, that help deter insects, you know, because they create these secondary metabolites that you've been hearing about, like and alkaloids that, you know, insects think are nasty. Okay, so interesting, right? So that's my example. And then the third thing are the links, right? So this is where you all come in. So you can think about, you know, arbuscular mycorrhizae can sometimes have an endophytic form, especially when they have, you know, part of them are inside a root. How can we then use that? And people have been thinking about and trying to use mycorrhizae, for example, with plant roots for phytoremediation and things like that. How could you then use that to create certain types of materials? I don't know, because I'm a couple degrees separated from bioengineering, but that's, you know, where you all come in. So those are the sort of three things I wanted to talk about.
Thank you, Sharifa. And just can I ask you to follow up on that? Can you define endophyte and contrast that with the term mycorrhizae?
Absolutely. Okay. So an endophyte is a fungal endophyte. I'll be specific because there are bacterial endophytes too. But here we're talking about fungi. So is a fungus that lives symbiotically with the plant and does not cause harm to the plant. And it's, I'll tell you, the frontier of science right now, because you know, we're learning about their functions. Sometimes they're sitting there like, we don't know what you're doing. And you can learn about it through microbiome work, metagenomics, things like that. Mycorrhizae, okay, are a type of fungus, okay? And they often associate with roots, and they help increase the surface area of the roots. Think of a sleeve on your arm. The sleeve would be the mycorrhizae, and your arm would be the root, and it would help you sequester things like water and phosphorus, okay? And in return, the mycorrhizae have a home, you know, so it's very cool. It's a symbiotic relationship, but it's specifically a mutualism, okay? Some mycorrhizae can be endosymbiotic, but there can be free living fungal endophytes living inside of plants that we just don't even know what their functions are. Maria, did you want to add anything to that? Okay. Thanks, Jen.
Okay, yes, thanks, Sharifa. So I'm gonna walk around because I'm antsy. So I want to bring this back around to biomaterials, fungal building materials. And I'm going to ask the panel and the room, really, whether or not it's realistic to imagine. We're going to start at the very beginning here, very simple. Whether it's reasonable to imagine a fungal building material with a fungal co-culture. So we're talking about a plant. So we're discounting the plant, by the way, which has made the substrate. And now I'm just asking, is a material that's made out of two different fungi potentially better than one that is made out of just a single fungus? Carolee, I can see you.
Yeah. So I'd just like to talk a little bit about what we know about fungi, because I think you actually can't get something that isn't more than one organism to be a building material. Yeah. And so
James loves that answer.
So if I will tell you a little bit about what we know about producing mushrooms. So mushroom production is done in a controlled environment. It could be in a room this big, if you like, where specifically composted materials that are composted using a two-tiered process or three-tiered process, depending upon how you want to think about it, to really get the materials in the right state, having the right microbial ecology that produces the organisms and that produces the fruiting bodies. Just an example, those microbes are necessary for producing fruiting bodies. If you sterilize the compost, you would not get fruiting bodies, okay? And there are organisms, Pseudomonas, which is one of my favorite bugs, that in fact will degrade toxins that are produced in the mycelium that prevent mushrooms from growing close together. So if it's producing this toxin, that toxin will reduce the number of mushroom fruiting bodies that you get, but the Pseudomonas breaks it down so the mushrooms can fruit close together. Okay, so there is a necessary set of microbiome dynamics that happen that allow fruiting body formation. And the area, and I'm going to just say the area of work that John and I and Kevin Hockett and Fabricio and Owen O'Connor are working on, Rachel started working with us too, is on the concept of developmental microbiomes. And in particular with mushrooms, there's going to be that. So you can imagine that these interactions between the mycelium and the bacteria, the mycelium with other mycelium are happening continually, and that there are microbial signals for growth and development. And so you may want a certain microbial signal to keep things from fruiting. You might not want fruiting bodies hanging off of your structures, or you might want them depending on what it is. And so I personally think, and I'll just say, as the current director of the Microbiome Center, I think that looking at what it is you want and then looking at the microbial community to help you get it from a community standpoint is really the frontier.
That's a very exciting answer. Kind of did, but I have more questions. So you're saying that to get fruiting bodies, you have to have a bacterium that is cleaning up the waste so that you can have dense fruiting.
That's in that system.
Right, in that system. So in hearing you talk, that made me think about something else. Could I have, so by the way, no one's answered my first question.
Yeah, we'll get there.
So if someone has an answer, just chime in.
So should it be in co-culture is what you're saying?
What I'm saying is, do we know enough about mycelium to understand whether or not a co-culture could have better materials properties? I think the answer is no, but I'm asking the question. Okay, we don't know. Okay, here's my next question. Could you have, now I'm thinking about carbon cycle, could you have a material in which you have a photobiont or a phototroph that's harvesting sunlight? Because really, here I am, the planetary ecologist. That is the only really actually sustainable source of energy, is the star. So the starlight is, ideally, we would incorporate an organism that can harvest starlight. can you have, so now this is like, can you make a lichen? Can you have a bacterium that's making cellulose from glucose that's made by the cyanobacterium that is then, turned into a building material that's bound together by the fungus? Like, could we make that? Any reason why we could not make that?
I would guess that there are combinations like that that exist in nature that we haven't looked at. And that's primarily because we, as you said, we've studied individual organisms, we've isolated them, put them on petri dishes, and look what they do in that artificial system, right? But there are all different kinds, there are so many different kinds of interactions, and we are just beginning to piece them apart, right?
Right.
And
Right.
so
So I guess to be even more sort of blunt, would NSF fund someone to go try sticking bacteria and archaea and algae together with the focus on building materials? Or is that just too far out? Is that too far ahead?
I love the idea because it's very sci-fi. Right? But I wonder, because the scientist in me is like, too many variables. So because we know, especially with fungi or even lichen, that you have to be very careful, because if you change the environment in any way, that can change the substrate, it can change their reproduction, they're alive, they're just not a piece of plastic, right? So it's very hard to predict. I think it's more interesting to think about what happens here on Earth, maybe not as much in the Petri plate anymore, because we know what happens in the Petri plate, right? But to make predictions based on these communities, how they might respond to extreme temperatures, to extreme environments, and then to environments that aren't as extreme, and how that might affect their traits. Traits, the communal traits, but also the traits individually. I think that would be interesting, but you might want to focus on a couple things. What do you think? Oh, yeah.
Can I go ahead? Or
go, James.
sorry, I wasn't sure if you could see my little hand raised. So, I do think actually there's a lot of work that is being done on replicating a lot of the attributes that we have from the lichen symbiosis, because really, a lot of the stability and a lot of the properties in terms of stability and I guess in the case of generating energy or what have you from algae, a lot of that is sort of what the lichens are designed to do. They're kind of unique in a lot of those ways. And so there is actually a lot of research that's being done actively on trying to sort of create de novo, synthesize lichen-like structures, right? Biofilms and all sorts of stuff like that. But the other thing is that, these organisms are already doing these things. And the thing that we are at the moment on the verge of, I would argue on the verge of understanding, is sort of the actual basis for allowing that symbiosis to happen, allowing the, we kind of understand the pathways involved in signaling for whether or not something is a compatible or not compatible, member, partner or something like that. But I really think that once we figure out, especially across a broad sampling of species, because remember, lichens they're a lifestyle, right? They're not a monophyletic group. So it's a lifestyle that's arisen independently, been gained and lost multiple times in numerous groups of fungi. So the question is sort of, are all these different fungi that are not necessarily related to each other, are they arriving at their relationship with these algae or whatever their associates in the same molecular manner, or is it different? And can we then use that to manipulate a system to build what we want? And I really don't think that's that far away, at least in terms of characterization. We have the technology to characterize these things. It's the expression and doing things in other systems that's the challenge.
Thank you, James. If I could just ask you to add on, is that being done in academia or in industry, or where is that work taking place?
To my knowledge, that's in academia. I don't know of anything that's being done in industry so much.
Okay, thank you. We've had a question back here, so I want to let this person speak, and then we'll take more questions as they come.
I was actually just going to comment on your question about the consortium. Hi, everyone. I'm Katie. So right now at the Curtis Lab here in chemical engineering, there's consortia like the one that you're describing, but for biofuels. And I think that the question becomes, can you have things that grow well together with their requirements and on the same time scale? Because mushrooms, the mycelia will propagate, I guess, at a certain time scale. And if you have a bacteria that's trying to also live in the same consortia, it needs to, have the same like oxygen, carbon availability requirements, whatever, so that they can work together. So the problem with or rather what the biofuel question is answering is Clostridium is breaking down cellulose and lignin in straw, and then an algae is taking those simpler sugars and converting it into oil that can be used for biofuel. So if we could do something similar to that, where we have bacteria breaking down the hay or whatever, and then also making the mycelial building blocks, like I think there's a lot of work being done in biofuels that could maybe be applied to biomaterials.
Wonderful comment. Thank you very much, Katie. Katie? Yeah. Okay, we have multiple questions. James, you still have your hand raised. Is it newly raised?
Yeah, I was just going to reply to that kind of with, I think one of the interesting things, again, is that using these lichens as a system, they've kind of already done that, right? They've evolved ways to essentially shut down the physiological activity and manage the physiological activity of the other organisms that are inside of them. So they are, physiologically inactive, as are all the things inside of them as much as they can be for protracted periods of time, and then they become active at certain periods. And so knowing the requirements that they need to become physiologically active, you could control that, and you certainly could manipulate that to your advantage. So, using lichens themselves or the ways that they have devised to do that for these other organisms, like that is a solution that in some ways has been solved, at least in nature. I'd be curious to hear about that in other fungal systems, because I don't know very much about that beyond my corner of the universe.
All right, thanks, James. Vinay?
Hi everybody, my name's Nick. I was gonna circulate back to the first question about a co-culture when we're doing like biomaterials. So my experience is with agaricus also, as Carolee talked about. And if you look through like the growing of agaricus, there is a co-culture the whole time with bacteria, but also with fungi. But that's a secondary decomposer, whereas the biomaterial fungi that you guys are proposing to use as a primary. So I guess you would have to find a symbiont or somebody that would work well with that type of fungi in order to do a culture like that. Yeah.
Wonderful, thanks, Nick. Vinay?
Not really a question, but maybe we talked about compartmentalization in science, like how we study one thing at a time. I think when we make our materials, fungal biomaterials, the problem is like we start by sterilizing everything. The substrate is sterilized, making sure that nothing else is living there. And then we move to the growth room. I mean, in our practice, it's in the Mushroom Research Center, the growth rooms that are clean and sterilized, where we inoculate that clean material with the fungi species that we want. And the goal is to make sure that only that fungi species grows and it like colonizes that substrate. But certain times, there are other environmental fungi in the room that, typically trichoderma can, enter the substrate. And when there's the contamination, there's no mycelial, there can be mycelial growth, but there's an ecosystem and there's a fight in that substrate. And the reason why we wanted to have this panel is exactly to discuss these, like, can we make biomaterials by creating an ecosystem instead of relying on one single fungal species? And can that also help maybe fight against mold? So can we find symbionts that will help the fungi thrive, but also fight against the other fungi species. So I know also that there are few studies in the literature that explore how we can, by combining different living organisms. Again, I'm an architect, so I'm just trying to understand all this literature too. There are also studies that explore how like creating a heterogeneous system can result in like higher mechanical strength, for instance, not just relying on one type of fungi, but a combination. But there are very few at this moment, but I definitely see a potential there. So I wanted to hear what you guys think. I mean, you already said what you think, but I just wanted to give that perspective.
Yeah.
Thanks,
so
Benet. Go ahead,
maybe
Carolee.
I'll take a stab at answering that.
Great.
So Benet, actually, this would be a perfect experiment to look at as you're growing. So I would guess that your materials are not sterile. I would guess that sometimes you have a pathogen of your fungus that comes in, but probably you have bacteria and fungi co-cultured with that, even though you've tried to be sterile, just because of the nature of what you're doing. And there's actually funding in the Microbiome Center that you could apply for. It's called Give a Dog a Bone, $5,000 of sequencing that you could do some sequencing to see who's there. Okay. And then the Microbiome Center, through their dog, because you're giving the dog a bone, their data analysis working group would actually help you analyze that so that you could figure out who's there. And then the question is, can you manipulate that to actually either make it stronger, make it grow faster, inhibit the trichoderma that's killing your mushroom material? So I think there are lots of questions related to what you're doing now. The more difficult piece is, where do we even begin to think about combining different organisms? I would say you start in nature and you start looking at, you could look at microbiomes of the kinds of organisms that you are trying to culture in a sterile environment and see what organisms are associated with them. There's some who's in there, but what organisms are associated with them. And that would be I think that would be the place I would start. And that is NSF funded.
There are companies that produce mycelium-based materials in like industries that produce mycelium-based composites, but they don't really disclose the mixture. But I don't know, but I heard from someone that their compositions are also not homogeneous. They were able to backtrack what's inside and found that there are different types of yeast working together with the fungi, resulting in faster growth and like more resistance against mold. I just wanted to say that, but I think that's that's what's needed to take these research also to the next level.
And there's some resources for you here on campus.
Great. That's great to hear. Sharifa, were you going to chime in?
I was just going to say that I think it's also important to think a little bit about sort of, like the method in terms of, you're saying you want to create an ecosystem, right? And sometimes you can look to ecological theory for that. And you can say that, you have all these different, you have a yeast, you have some bacteria in there, you have some fungi in your mix, whatever it is, that you're trying to work with. And, it might be very difficult, for example, to mimic what's in nature if you're going to, and you should start with nature. I think you should definitely start with nature. But the cool thing is that all organisms, have functions, and there's this concept of functional redundancy, where certain functions you can find in different organisms, and some organisms are easier to grow than others. So, and you can take either a phylogenetic approach, where you look at closely related organisms, and the idea is that sister taxa, or like, think about your own families. You probably look more like your parents than your friend looks like your parents, right? Because you're related. And the same thing with organisms, they'll have those traits. I'm sorry, it's been a long day.
She's laughing at my face.
It's a good example. You won't forget it, right? But so we can have these, if you're looking for, lignin degradation again, just as your function, maybe there's certain organisms that grow really fast, that are very common, and that work well with others in your ecosystem. And so that is part of the selection process you would want to put into a grant, I would think, like how you screen, because the screening is like the hardest part.
Right. Thank you, Sharifa. I'm going to return to screening in just a moment if we have time. But in the meantime, let's take a question from the chat.
Question from the chat, again related to mold. Can the alkaloids that endophytes create help deter mold growth? Could the gene from these endophytes be transferred to white rot fungi through genetic engineering?
So excited about the idea of you giving a dog a bone. that I was just like, ask the question.
Okay, the question is, can the alkaloids that endophytes create help deter mold growth? Could the gene from these endophytes be transferred to white rot fungi through genetic engineering?
I think, could they? Yes. Would they be safe? Who knows, right? It depends what alkaloids you're talking about. So whether you'd want to or not is another question if it's for a food product, right? If it's not for a food product, that's a different kind of question. Did that answer that? Getting a dog bone. I already sent you the email. The grant is due on the 15th. It's short.
All right. All right. We're gonna get Benet hooked up with dog. Absolutely. Dog bone.
We're gonna get her a bone.
We're gonna get the dog a bone. Okay. We are going to. At what time are we going to adjourn? 5:15, I think is. So we have a few more minutes, 12 or so, to talk about whatever you want to talk about. And so I encourage you to ask whatever kinds of questions you might have. And if you don't, I have a whole list. So don't think you're going to leave if you don't have a question. You're here for the duration.
Could I just talk about the developmental microbiomes?
Absolutely.
So Monica Medina, who is in biology, organized a group of people to think about writing an NSF grant. And we did. We wrote a pre-proposal for an NSF center. Yeah. And so people looking at developmental microbiomes for all different kinds of things, everything from jellyfish she works on to fungi, which Fabricio and a bunch of us were involved in, and then myxobacteria that are developmental organisms, lots of different things across the kingdoms, right? So this is a fundamental, what role do microorganisms have in development throughout the tree of life, right? And so when we're thinking about materials, this is one of those keys I think that we need to think about is what, and Benet is trying to get at that, but what is it, if you're trying to grow a material, what are those organisms that are going to give you the right consortium? I think it's the same question you're asking, but that fundamental question about, are there developmental microbiomes or things that increase growth and development, in addition to, just saying what consortium would allow them to be sustainable by feeding themselves, just thinking, what are the organisms that are involved in, just allowing them to grow? I'd love to see Monica bring that group back, that rock band, for a second try, just because it's, we got good at singing, but we weren't playing the drums quite so well.
Any other questions? Or you're also welcome to email us or set up a time. I'm totally volunteering, Dr. Bull and
Yeah.
James.
I think Sharifa, you had started there too. to, bio-based materials and composites, right? That's where I'm coming from as a mechanical engineer and material scientist. And there's something that I heard multiple times, starting with this morning, and then, from Elise, and then Tamash, James mentioned it, and then you, when you talked about the three, I don't think you called them pillars, that was Tamash, forgot what you called them, but you talked about framework, example, making a link, which I love. And you picked up again on this thread, which is the function, right? If we can't use the thing, and I'm paraphrasing James, think of using the function. It is basically what we talk about, as engineers, when we're talking about multifunctional composites, right? So when we're talking about multifunctional composite, we have our ecosystem, which is multiple materials that are coming together, that we're forcing them to be together. When we fabricate them, they don't necessarily want to be together. They wouldn't normally, connect and give us good properties if we didn't force them through chemical means to do that. So the reason I'm saying all this to say, getting back to bringing multiple fungi, together, or fungi and bacteria, etc. If in some ecosystem, we have these multiple species types together, where, I would like one of them or two to be, capable of being activated so that they can feast on the other material at end of life, for example. That's their function. Their function is eating the whole thing and breaking it down, so that we are truly sustainable, at end of life. And then a couple of them are actually what is going to help this ecosystem and help their friends grow and give us, a dense material that is able to have good mechanical properties.
I love that. Okay, so here's an idea. So when you were talking, I was thinking, wow, we have to think about, if you want, you said you make the materials work together through chemical means. So how could you do that with a bunch of microbes and fungi? Maybe you could modulate the environment. You could change time and space. Time is you introduce them at different times, and space is you compartmentalize them, maybe within a lichen, maybe into something else. I don't know how, but just thinking about how you modulate the environment such that, these microbes can interact. But remember, microbes are fickle things. So, because they're alive, so you can't always predict what they're going to do, unlike, a dead material, but it's not a non-alive material. So I don't know. I just thought of that, but at least it's a starting point to think about modulating the environment. I don't know. What do you think?
I think it's a great. I'll say that's one of the other pieces, and I think James got at it in his presentation, and I really appreciated you talking about colonization and time and space, right? It's an important piece of what we do. So for example, Benet, for your work, do you add your inoculum all at once? Do you do it in one place and hope it grows in a direction? Do you inoculate across? All of those things could have impact on, it could be even strength, but it could be impacting the colonization by other organisms. And so spatial temporal interactions are really the frontiers of microbiome science.
Well, and it's quantity as well, right? I mean, it's not just, it's sort of time, place, and then also, I guess, abundance is maybe not necessarily the way to think of it, but there are so many processes that have a threshold to them that has to be reached before it will happen, before whatever needs to happen is going to happen. And that's really one of the, getting the thing at the right time, doing it at the right time and in the right place, but then also making sure that there's the right amount, or exceeding or not exceeding whatever the threshold is really critical, or keeping something at near close to threshold. So I think it's really abundance and understanding those sort of thresholds as well, tipping points, if you want to think of them that way.
Yeah, and then exactly, and also think about then the biology of those organisms. because they may all have different
Yeah.
times, you know, or they
So,
may be able to just clonally
I'm going to
propagate.
pick right up on that, Sharifa, because, and I'm going to put words in James's mouth, because I heard James talking about chemical handshakes that are what it takes to get partners together, right? So I think that understanding signaling is really important, because if we provide the signal, people could get together even if they weren't the ones that originated the signal, right? So if we understand the signaling part, I think we really need to involve some chemical ecologists in this community building exercise. And we have some of those here at Penn State. So
That's how you throw a party, right? Not everybody plans it, but they show up. They get the signal.
right, they get the signal. Okay, so we I think that we could adjourn with the panel and move on to our next growth phase, right? Okay, which James, sadly, you will not, you know, you will not be able to be physically here, and all of you on Zoom. We are going to move into a poster session and a reception. And so any last, I think we're going to make a few closing comments here and then move on into that next phase. Zubeida, is there anything you'd like to communicate?
Which is exactly what we started to do with this awesome panel now, right? We really wanted our discussions to be focused on how do we move forward as multiple interdisciplinary teams. What Carolee was talking about with Monica Medina's effort, that's another outlet that I hadn't mentioned this morning, which is the science and technology centers, which are extremely inter and multidisciplinary. And that's something else we might want to think about.
And let's just thank our panelists before it gets too late here. Thank you very much.
Thanks, y'all.
Thank you. Thanks, James.
Yep.